Hi everyone and welcome. Today we're diving into human T-lymphotropic virus, and we're going to look at it specifically through the lens of what it means for us, for medical microbiologists right here in the Netherlands.
Just a quick heads up before we jump in. This entire explainer was put together using a Gemini deep search. For any of you who want to go deeper and check out the primary literature, there's a complete list of sources waiting for you in the linked Notebook LM document.
All right, so here's our game plan. First, we'll get into the virus's backstory. Then we'll unpack the diseases it causes. After that, we'll look at how we diagnose it here in the Netherlands, explore why it's so tough to treat, and finally, we'll wrap up with prevention and control.
All right, let's kick things off with a bit of history. We're talking about a pathogen that's a really big deal globally, but, well, it's something you almost never see in our local Dutch context.
I mean, it's really hard to overstate just how important this virus is in the history of virology. See, when it was identified back in 1980, human T-lymphotropic virus was the very first human retrovirus ever discovered. It really paved the way for so much of what we now understand about this whole class of viruses.
And when you look at the global picture, the numbers are pretty staggering. It's estimated that somewhere between 5 and 10 million people are living with the virus, especially in places like the Caribbean, Japan, and sub-Saharan Africa. But this global scale is just a world away from what we see here at home.
Yeah, because here in the Netherlands, this virus is a real rarity. You're almost exclusively going to find cases in individuals who've come from these endemic areas. And there's a really notable link to Suriname. And this one epidemiological fact, it shapes our entire public health approach.
Okay, so how does this thing actually work? What's going on under the hood? Let's zoom right in on its classification, its structure, and of course, the really severe clinical syndromes it can end up causing.
To really get why this virus is so dangerous, you've got to look at its family tree. It belongs to the Delta retrovirus genus, and this is a crucial distinction. See, unlike lentiviruses like HIV, which we often think of as just destroying cells, Delta retroviruses are known for their oncogenic potential. They can literally transform host cells and cause cancer. So in a way, its taxonomy tells you exactly what kind of trouble it's capable of.
And if you look at its structure, it's pretty much a textbook member of the Retroviridae family. You've got this spherical particle wrapped in an envelope about 100 nanometers across. And inside that envelope, which by the way it steals from the host cell, you've got the icosahedrocapsid doing its job, protecting all the important stuff, the viral RNA and enzymes.
Now, it's important to remember that only a small percentage of carriers actually get sick. But for those who do, the outcomes are just worlds apart. On one side, you've got adult T-cell leukemia/lymphoma, or ATL. This is a full-blown cancer of CD4 plus T cells, and it often takes decades to show up after someone was infected as a baby. Then on the complete other side, you have HAM/TSP. This isn't cancer, it's a chronic inflammatory disease of the spinal cord, and it's all driven by the body's own immune response to the virus. And this can pop up much, much sooner.
So if we zoom in on ATL for a second, the clinical subtype makes a huge difference for the patient's prognosis. While the smoldering type can be slow-moving, the prognosis for the aggressive, acute, and lymphoma forms, well, it's incredibly grim. We're talking a median survival of just over a year, and that's with treatment.
And the picture for HAM/TSP, it's one of this slow, relentless, progressive disability. It's all driven by that chronic inflammation in the spinal cord, which leads to these absolutely debilitating symptoms. Think slowly progressive stiffness and weakness in the legs, terrible back pain, sensory disturbances, and significant issues with bladder control.
So with these incredibly severe outcomes on the table, how do we actually manage this virus in the Netherlands? Well, the truth is, our entire approach is really laser-focused on one thing: prevention.
Okay, so the diagnostic algorithm we use is super clear and logical. Step 1 is screening. You start with a highly sensitive test, like an ELISA or a CLIA. If you get a hit, you move to step 2, confirmation. This is where you bring in a highly specific test, like a Western blot. And for step 3, to clear up any gray areas, you know how common those indeterminate results can be in a low-prevalence setting like ours, we go for the final word, definitive molecular testing with real-time PCR.
Now, if you're a clinician and you go looking for Dutch treatment protocols from, say, the RIVM, you're not going to find much. And this quote really gets to the heart of why. The fact that we don't have these specific national guidelines isn't some kind of oversight. It's a direct result of just how successful our prevention strategies have been at keeping this virus so incredibly rare.
And here it is. This is the absolute cornerstone of our public health strategy, the mandatory screening of every single first-time blood donor by San Quinn.
This one policy, it has been unbelievably effective at protecting our blood supply. It's virtually wiped out transfusion-transmitted infections in the country.
Okay, but what about the people who are infected? Why is treatment so unbelievably difficult? Well, to get that, we have to go back to the virus's fundamental biology.
This brings up a really central and, let's be honest, a frustrating question in this field. We have antiretroviral drugs, and they work great against the virus in a petri dish. So, why do they pretty much fail to reduce the viral load in patients who have a chronic infection?
The answer, in 2 words, is clonal expansion. This is the whole ballgame. See, in a chronic infection, the virus doesn't really need to bother with infecting new cells all the time. Instead, its main trick for sticking around is to make the host T cells, the ones that already have its DNA integrated into them, divide. So the virus just gets copied along for the ride every time the cell divides. This completely bypasses the enzymes like reverse transcriptase that our drugs are designed to hit. It's a clever and frustrating loophole.
And that biological challenge, that loophole, it just highlights why prevention is so absolutely critical. So for our final section, let's look at the public health strategies that have been a genuine success story here in the Netherlands.
Globally, the prevention playbook is pretty straightforward, and it works. It all comes down to interrupting the 3 main ways this virus gets around: parenteral, sexual, and vertical. So that means robust screening of blood and organs, promoting condom use for sexual transmission, and of course, advising infected mothers to avoid breastfeeding to prevent passing it to their children.
And this right here is a perfect example of how public health policy is tailored to our local situation. The RIVM standard perinatal screening for infectious diseases, it does not include human T-lymphotropic virus. And that's not a mistake. It's a decision based on solid epidemiology. The prevalence is just so incredibly low that a nationwide screening program would not be cost-effective at all.
Which leaves us with this final lingering question. Since our prevention strategies have basically halted transmission right here in the Netherlands, can we call this a solved problem? Or is it more of a persistent threat, hiding in plain sight within a very specific group of people, just waiting, sometimes for decades, to finally cause disease?
It's definitely something to think about. Hey, thanks so much for joining me for this explainer. If you want more expert reviews like this one, please do subscribe to the channel, and we would genuinely love to hear your thoughts and any questions you have down in the comments below.